Okay, thanks. So yeah, as I said, feel free to hop in if you have any questions, since we're a small crowd. So, but I want to start by saying that I'm excited to share this work in the context of a bioinformatics seminar, just because I think I spend a lot of time talking about the applications and don't get to geek out about the kind of the bioinformatics details as often. So this is my chance to do that. And then I want to say real quick that this whole project is very much a team effort by myself, Michael Altermott, who is a recent graduate, and Tatiana Dobriva, who's also a member of Viviana Gradinaro and Matt Thompson's lab, which this work is a part of. So to start, I want to give some background on AAVs. You all have probably heard about AAVs or used them, or you know someone that uses them. But just to frame the work a little bit differently, I'll give background in a specific context. So AAVs are really cool. They're small viruses. They don't integrate into the host genome, and they have a relatively low immune response relative to other vectors used for gene delivery. They also have a broad native tropism, so they hit a lot of different cell types just from their wild type variants, and they hit both dividing and non-dividing cells. And I think for me, one of the things that's coolest about them is that they are heavily engineerable. So they have about a 4.7 kb single-stranded DNA capacity, and you can easily trick them into carrying whatever gene target that you want by when you manufacture the AAV, you deliver your gene of interest to like your producer cells, your hex cells, and you flank it with these inverted terminal repeats, and the AAV will think, oh, that's my gene, I'll take it, and I'll deliver it for you. And for all these reasons, AAVs are one of the major players in the gene therapy field, making up, as of 2019, about a quarter of all gene therapy clinical trials. But I think it's also important to note that they are not the only player in the space, and that the work that I talk about today, I think, is generalizable to gene therapy delivery in general. So I like to think about this from the perspective of, if you're a gene therapy researcher, or either for clinical applications or for research applications like behavior research, how might you think about the viral vectors that are out there? From your research perspective, you think about what you want to deliver. Are you rescuing a gene? Are you editing the gene with CRISPR? But once you figured that out, which is probably the bulk of your research area, you need to figure out how to deliver it to where you want it to go. And you have some criteria. You have, what are the cell types that are implicated in the disease or behavior that you want to deliver to? And where do you want to avoid delivery? Either organs that might cause some sort of immune response, or cell types that might confound the research that you're doing, if it's like behavior research. And so you have a lot of choices when you go to decide how to deliver your gene. You can choose the vector. AAV is one, but there's also adenovirus, HSV, lentivirus. And then even once you've chosen the vector, there's this kind of triple effect of what capsid, what specific variant do you choose? What delivery method do you choose, whether it's systemic or local? And also, do you make any modifications to your cargo that will change how your gene is expressed? And for these two things, for capsid design and cargo modifications, there are a whole bunch of options. Some of the high throughput methods that have come out, such as the Cree-based method from the Gradinaro lab, can produce millions of variants by introducing these libraries of mutations that then select for expression in specific tissues. And there's been similar work done for promoters and enhancers, and also using RNA-based screens and selection methods. And so as a gene therapy researcher, you have a lot of cool tools now, like the brain variant that came out a few years ago, PHPB and PHPEB, that hits brain-wide, as well as one that came out last year from Priya Kumar in Viviana's lab that hits specifically vascular cells. But there's a problem. There's a bottleneck here, which is as a tool developer, when you go publish these papers of a new variant that you've found, you have to pick which variant you want to characterize, and then you also have to pick which particular cell types and tissues that you're interested in. And every new choice is more lab work and more animals. And so there's this kind of not antagonism, but like this different perspective from the vector developer versus the gene therapy researcher, which is the vector developer, they can make their own vectors. They probably want to target the biggest use cases like brain-wide expression. And that's what they will include in their characterization efforts. And so you could imagine a vector developer saying, hey, I've got virus A that delivers to cell type A, virus B hits cell type B, and you've got this whole pool. And then gene therapy researcher comes by and says, okay, what about my special cell type omega that you didn't test in your paper? How does it do? And as we know from single-cell RNA sequencing, every week there are more and more cell types in different tissues and organisms coming out. And so the likelihood that you have characterized the one that an eventual researcher wants is quite low. So what you ideally would want is an unbiased readout where you can deliver vectors, preferably multiple, and then get a vector report card of how does this vector perform across the entire cell types in the cell type hierarchy. And that is the where single-cell RNA sequencing comes in. So in a nutshell, single-cell RNA sequencing is you take a tissue from an animal, you run it through a machine, and you get this gene by cell matrix, which what you can then do with it is you can figure out what kind of state each cell is in, what type of cell it is, and all the genes that are expressed in it. And what we want to do is make a slight modification to this protocol where you take a virus and inject it into the animal before running the procedure. And then in your gene count matrix, you can then get for each cell what level of virus expression that cell has. And ideally, even better would be multiple viruses that you can then add multiple rows to your gene count matrix of all the different viruses you've delivered. So in a nutshell, that's our pipeline, and that's our strategy and the goal of this project. And so now I'll walk you through the details of where things went wrong. So we start by taking a pool of viruses and injecting them into an animal, and these viruses have some kind of distinguishing barcode in their cargo. We then do the typical single-cell dissociation procedure, get single-cell suspensions, and then run it through a droplet-based single-cell sequencing method like what we used was from 10x Genomics. And what you get at the end of the single-cell sequencing pipeline is a full-length cDNA library of all of the RNAs that are expressed in your cells. And you can figure out which RNA came from which cell because a cell barcode was added during the droplet sequencing procedure. And what we also have is the viral cargo transcripts, which now have this barcode or this distinguishing sequence that tells you which variant each specific virus barcode is associated with. But here's where the first problem comes. So one of the first steps in the single-cell procedure for preparing your library for sequencing is a fragmentation. And what happens during this fragmentation is you only get the region of the gene that's near the poly A tail. So you lose the distinguishing variant barcode region. And this will happen because a lot of the stuff that's near the poly A tail is usually common between a lot of different genes that you might deliver. This is where regulatory elements go, like the WPRE. And so the stuff that distinguishes the virus, if you have it distinguished by different cargo, is way outside of the reach of post-fragmentation. And if you were to put a barcode over near the poly A tail, that would also be too far away from read 2, and it would be potentially inaccessible from read 1 because it has to go through the poly A tail. So the solution that we came up with is to amplify the viral fragments with a primer that binds right before whatever distinguishing sequence for the virus is. And what you then get, what we hope for, was what you then get is you get your normal cell by gene matrix, but then also you get a separate sequencing data set where for each cell barcode, you have which virus transcript is in it and how much. And what we learned from this is that this, in general, this works. And although you may lose identifying regions during the single cell workflow, you can then amplify and sequence them. And we found that this worked for pretty large fragments, up to almost 900 base pairs. So that's the main summary of that strategy. And so we decided, let's go do our first validation experiment. And the experimental paradigm was, we had 4 animals, and we injected 2 of them with variants individually, and then we injected 2 animals with co-delivery of 2 variants that were carrying 2 different fluorophores, amnion green and TD tomato, so that we could take half the brain from each animal and quantify it via imaging, and take the other half and quantify it via single cell sequencing. And we got our imaging results back, and it was great. We could see what percentage of cells were transduced by the 2 different variants, and the patterns matched what we had seen before. These 2 were known variants, PHP-EB and CAP-B10. And here's where the next problem came in the single-cell RNA-seq data. So when we looked at the viral transcripts in all of the different cell types that we detected, most of the cells had almost 100%, almost 100% of the cells had some level of viral transcripts in them, and they had extremely high level of viral transcripts, even cell types that you would never expect. And we just know from imaging and anecdotally from work in the lab and from previous literature that there's no way that these viruses hit 100% of just about everything. So we wondered if there was some kind of artifact that we were introducing during this amplification process. And luckily, for the 2 animals where we didn't have multiple variants, so we didn't really care about this barcode region, we could look in the transcriptome to see what these what this data looked like without the amplification. And what we found was, what you see here on the x-axis is the number of viral UMIs based on our amplified data, and each dot here is a different cell barcode. And on the y-axis, you see the number of viral UMIs in that same cell from the transcriptome data, not amplified. And you can see that there's this very clear artifact that shows up where there are a bunch of cells that have very high UMIs in the amplified data that have almost nothing in the transcriptome, suggesting that this is some kind of artifact. And when we look at the raw sequencing data, and this is, I'm going to be showing a lot of histograms, so it's sometimes hard to keep track of what the X and Y axis means. In this histogram, the X axis is the read count, how many times you read the same molecule in the sequencing data. And on the Y count is how many molecules we see with that high of a read count. And you can see that most of the transcripts that we detect in the amplified data have very low read counts, on the order of single digits. And if you zoom way into this histogram on the right here, there looks like there are multiple distributions. And from what we know from PCR, we hypothesized that there were 3 different sources of data in these histograms. The first, this nice peak over on the right, is our real amplified transcripts. And then over on the left here, we have 2 different sources of error. One is from mutation errors and sequencing errors, where there's just a single base pair. And these can be modeled by a binomial distribution, because each mutation is exponentially less likely than the previous one. But we also have this more complicated template switch type error, which is what we think explains this kind of stretched out error distribution that can't be modeled with a binomial. And I'll go more into what the template switches are later, but in short, know that we identified 2 distinct sources of error. And the benefit of being able to fit 3 different distributions, one that you know is real and 2 that are errors, is you can, once you fit it, you can use Bayes' rule to then calculate, if I have a certain read count, what is the probability that that transcript is real, as opposed to coming from this noise. And so this was exciting when we took this data and we ran it through this fitting procedure, and then we compared the amplified to the transcriptome counts. You'll see that the artifacts are completely gone, and that there's almost a perfect correlation between the now amplified corrected counts and the counts that we saw in the transcriptome. with the exception of, it looks like maybe this one or maybe a few here. And I suspect that these are transcripts that were lost during the amplification procedure that were then unable to be recovered. So this was great. We now had a function that could take these reads from amplified data and convert it into a probability. We thought we were good, but then something weird happened. When we looked at these fitted models in different cell types, we realized that it seems like these probabilities of template switches seem to be different for different cell types. So if you fit the same model for the same cell type, you'll get a wrong picture of the estimate of error. And so we were seeing cell types that had strangely high number of transcript counts that didn't really match with the sequencing or with the imaging data, leading us to believe that this wasn't a fully solved problem. And when we dug into this a bit further, we realized that what seemed to be happening was in oligodendrocytes in specific, there were some off-target sequences that had a high degree of homology with the viral amplicon. And it seemed to be that this was a oligodendrocyte-specific gene. And so that's why we were seeing it only in oligodendrocytes and not other cell types. And so what's likely happening is during PCR amplification, we have our fluorophore, our M neon green transcript, and we have some off-target transcript, like from oligodendrocytes. And when the polymerase goes and does its amplification, because there are regions of commonality between these two, it will flip in the middle and associate the wrong UMI and cell barcode with the wrong transcript. And this happens at different rates depending on what genes are expressed in that particular cell type. So it's a big mess. And so we had a few different options. The first was, well, you could fit a different model now to each cell type, but that would be a pain because you better know your cell types. As soon as you get one wrong, you might be making claims that those cell types have higher expression when it could be some type of artifact. Another alternative is to switch to a droplet PCR method, which would be the, I think the default wet lab solution to this problem. But to minimize wet lab work, I tend to default to computational solutions. So the idea was, why can't we use the transcriptome? So going back to this picture, you have all these reads that are from the transcriptome, and then you have the amplified reads. And even though we lose the variant barcode, what we can do is we can still identify which cell barcode UMIs are associated with virus. And then we can simply use the amplified reads as a lookup table to identify which cell barcode and UMI is associated with which viral variant. And that's what we ended up doing. So we didn't end up using the PCR fitting method, but I wanted to share it because everyone does PCR and it might be of relevance to other folks. But this lookup method seemed to be a lot better, and it recovered most. We didn't lose a lot. We only lose about 30% of the transcripts when we do this lookup table method, which is plenty for our application. So the next lesson here that we learned was PCR introduces artifacts and that you can correct them computationally. But unfortunately, this correction has to happen on a per cell type basis. So just to zoom out a bit, going back to the big picture. So what we've got now, finally, is we've got this first part of the pipeline down, and now we've got a count for how many viral transcripts we see in each cell for each AAV variant. And we can now go back to our initial validation experiment and see these biases in the single cell data. And the bias that we expected was confirmed. So S100 beta refers to astrocytes, and RBFOX3 is a neuron marker. And you can see here, it's just visually striking from the t-SNE itself that there's a lot more bluish in the astrocyte cluster for PHP-EB than there is for CAPI10. And similarly, there's a lot more blue relative to astrocytes in CAPI10 for neurons. But how exactly do we quantify this? The straightforward way might be, okay, any cell that has a transcript, we'll call that transduced, similar to how we do in imaging. If it's bright, we count it. If it's not bright, we don't. Because what we really want to know is not the expression rate, but what percent of cells are receiving the transcript and then expressing the transgene. And from a probability perspective, this is what is the probability that X is greater than 0. So why don't we look at RNA expression levels? Why am I talking about this probability greater than 0? And the reason is that different cells express different level of the delivered transcripts. There's variability in different cell types because of promoters. There's also overall level of activity. Some cells are just more active and might express more transcripts. And then there's also just cell size. Bigger cells tend to have more RNA. So is it really fair to have a threshold and cut off cells that might have low expression that's meaningful, as opposed to counting them? And as evidence of this, we see this very clearly in our astrocytes versus neurons example, where when you look at the mean expression level of astrocytes, it's about 7 transcripts per cell. However, 60% of astrocytes have expression greater than 0. Whereas with neurons, they have about 8 transcripts per cell, even though only about 35 to 40% of these cells are expressing the transcript. So if you were to base your claim about where is my gene being delivered most to on mean RNA expression level, you would conclude that this virus is hitting neurons primarily, when the truth is that in terms of how many cells are actually being hit by it, it's astrocytes. So this is why it's important to establish what is this probability X greater than 0 as opposed to these mean expression levels. And unfortunately, you can't just look at the raw data and say which cells have greater than 0. When we do this, we find that cells that we know have low expression are still showing up as significantly transduced. And there's no clear elbow or threshold here that we could identify as this is what makes things transduced versus not. And so what is this coming from? Why is it that cells that we know are not transduced are getting 50% of them have at least one viral transcript? And to answer this, we have to go back to how does droplet-based sequencing work? And to zoom into these little droplets that are going through the machine, what you have in each droplet is you have debris in addition to the cell and the bead that's in each droplet. And this debris comes from just during the dissociation procedure, you have cell chunks that become part of the soup that gets expressed or it gets included in a droplet that has cells, as well as just empty droplets that are part of the background. And so we needed a way to answer how many of these cells express the viral transcript given the level of background that we see. And so we were wondering, similar to other methods like the empty drops method, can we somehow use the debris as a way to inform what we expect to see in the soup versus what we see in the cells? So what we have is the distribution of debris droplets that we can get from looking at droplets without any cells. And what we also have is a distribution of transcripts in cells with debris. So this would be the probability of the cells plus the debris. But what we want is the distribution for the probability of just transcripts in cells, or specifically the probability that a cell has greater than 0. And unfortunately, if we model these things as negative binomials, there's no closed form solution to calculate this probability C greater than 0 given the two distributions that we have. So instead, we came up with an empirical method where we use the debris transcripts as an estimate of the background, and we then use the combined transcripts, and then we make a bunch of different guesses as to how much debris we think is in the sample. And what you can find is if you calculate the error of this combined estimate and what you see in your real data, you can find the level of contribution of the background that minimizes this error, which is our best guess at what percent of the cells have transcripts above background. And we were able to simulate this because we have distributions of the background transcripts, and we can generate infinite amounts of theoretical negative binomials for transcripts within cells to then sample from this theoretical C plus D and then use our method to recover the estimated transduction rate, which we found was in very strong agreement with the theoretical probability of a transcript being greater than 0. So that was great. And our lesson learned here, and this is, I think, a really important one for droplet-based sequencing in general, is that you cannot confidently call the transduction status, or even I would say the gene expression status, in any individual cell. You can't say, I know that this cell has this transcript because of these noise problems. But what you can do is you can take a group of cells and estimate what fraction of them are above the background, which is exactly what we need for our viral transduction case. So finally, now going back to our validation experiment, we have all the pieces in place, and we found that using the single cell data, that we found the same bias when we quantified it as a fraction of transduced cells. This CAP-B10 transduces far more neurons relative to astrocytes than PHPEB. And encouragingly, we also found a high degree of correlation within when we looked at the pairwise animals, within those cell types between the transduction rate as estimated by imaging and the transduction rate estimated by single cell RNA sequencing. So one thing that I've kind of glossed over is, we call these things neurons and astrocytes, but one of the key components of any single cell workflow is the cell typing pipeline. And in our neuron data, when we looked at our t-SNEs of cell types, it was pretty depressing. We always had this giant blob in the middle of kind of meaningless junk, which we assume was debris because of the fact that it had no expression of marker genes in it relative to all the other cell types. And so to type our cells and to also determine what's the difference between empty droplets versus not empty droplets versus droplets containing multiple cells even, we found out we can't use the kind of off-the-shelf solutions. So Cell Ranger V2 has this elbow fitting method that finds a threshold to cut off which is a cell versus empty. And unfortunately, that assumes a homogeneous transcript count distribution, which for heterogeneous samples like brain is definitely not a good assumption. And even an empty drop space method, like what's implemented in Cell Ranger versions 3 and later, they assume a homogeneous background distribution, which again, in the neuron data, because we have these cell chunks that are making up debris, is not a safe assumption. And so we had to come up with our own way of filtering out the debris and identifying what our cells are. And we realized we could use these marker gene expression levels directly as a way to determine whether something is a cell or not. And so a quick aside about marker gene identification. When we think conceptually about marker genes from a biological perspective, we want a marker gene to be a cell that is expressed in most of or many of the cells in a defined population. And you also want them to be uniquely expressed in that defined population. And you don't really care about the expression level. If you have a neuron that is expressing twice the level of marker gene as another neuron, you're not going to say that the neuron that's expressing twice the level is more markered than the other neuron. They're both neurons because they both express the marker gene. And so a lot of existing marker gene identification pipelines are based on this idea of expression rate, the number of transcripts per cell, instead of what is the probability of that gene being expressed. So we're back here again to the probability of X greater than 0 versus the expectation of X. And these are not the same thing. And they're not even rank correlated necessarily in distributions like the negative binomial. So mean expression, in my opinion, is not the right metric for determining whether a cell is expressing a marker gene. And as evidence, again, we saw that with the astrocytes versus neurons. But one of the cool things that we realized from SCVI, which is a package that lets you model your gene expression data, your single cell gene expression data, as samples from a learned negative binomial distribution, is that you can generate from these learned samples expected counts in your clusters of interest. So you can sample this posterior directly and get an estimate of probability X greater than 0. And it isn't limited to just the negative binomial distribution, because there is a complicated model that has learned the expression of these cells in your different your different groups. And so we think this is a lot better way for determining marker genes for debris detection. And coincidentally, you can also use the same exact strategy for determining marker genes for cell typing, which is a nice freebie. So we have this pipeline now that does clustering on a subset of the data, and it then determines which clusters have marker genes based on this probability x greater than 0. And you can then identify which cell types, or sorry, which clusters are multiplets because of expression of multiple marker genes, which clusters are debris because of expression of no marker genes, and which clusters are the cell types that you're interested in based on expression of their specific marker genes. And we use that to get a picture of the main cell types in our samples, the major 5 cell types: neurons, astrocytes, oligodendrocytes, and microglia. And the lesson that we learned here, and I really want to stress this, is the default cell calling methods in single-cell RNA-seq may not work for your sample. So look carefully. And this other point that probability of x greater than 0 is not equal to the expectation of x. And usually, from a biological perspective, we want probability x greater than 0, unless you really are looking for upregulation of genes as opposed to binary switches. So what we did now is we have this data set. We have these cells that are infected with 2 viruses. And instead of just looking at marker genes based cell types, we can also look at the whole hierarchy of cell types that are present in our sample. And we did this by building a model of our sample combined with the Allen Brain Atlas, which is a very well annotated data set for neurons, and use that to identify which cell types we have. And then we can, in one fell swoop, identify the transduction biases of these 2 variants in all the different subtypes of neurons in the brain. And what we found is, you'll see here that we knew that CAPB10 expressed more in neurons than PHPEB, but it seems that this difference in expression comes specifically from excitatory neurons, not inhibitory. And we went back just to make sure and validated this using FISH HCR for the specific genes that we identified in single-cell RNA-seq, and found the same pattern. And we also found this other subtype of cells, these VIP positive inhibitory neurons, that don't seem to get hit very high by any of the variants that we tested. So that was cool. The next step was, we want to scale this up. We don't want to keep using different fluorophores for every virus that we want to test. And so we developed a alternative cloning and barcoding strategy, where instead of having multiple viruses that deliver different fluorophores, that each virus delivers the same fluorophore, but now has a barcode that's introduced in the poly A region. And this basically switches the problem from before of the identifying region being over here, far away from the poly A region, to being over here, near the poly A region. But the pipeline is absolutely the same. Primer, amplify to recover your barcode. And we did this for this PHPEB and PHPV1, V1 being a variant that expresses highly in vascular cells. And what we found is, kind of expectingly, that if you look at the variability within barcodes with a single animal, it's a lot less than the variability between animals. And so the point that I kind of want to stress here is, biological variability is quite high with these different gene therapy delivery characterization methods. But if you can get a metric from a single animal and compare things within an animal, you're going to get more stability in your conclusions. And for sake of time, I'll skip over a few of the details here and jump straight into the finding from PHPEB versus V1. So when we repeated this experiment using both the barcode method and the cargo method, we confirmed this PHPEB versus V1 bias, that V1 definitely hits more vascular cells relative to other non-neuronal cell types. And we drilled down into the non-neuronal cell types based on known marker genes from the literature. And we found that this bias for PHPV1 comes specifically from endothelial cells and none of the other vascular cells in the brain. And this is important, for example, for researchers that are studying pericytes to know that pericytes might not be getting hit by this vascular specific variant. And then our final experiment was to go big and do a bunch of variants in a single animal. And so we have a new construct here, which is basically the same as the previous one, with some slight modifications just to make it easier for cloning so that we can get this barcode region incorporated into the plasmid easier. And we did an experiment now where we did 7 variants in a single animal. And unfortunately, one of the first things we found was that this new construct that we used, this UBC M-cherry, had significantly lower transcript abundance than the other constructs that we had used before, MNeon green or CAG-TD tomato. And also, it's not just that the transcript abundance was lower, but the cell type bias itself was shifted from the fluorophore alone, or not from the fluorophore, sorry, from the whole cargo, the fluorophore plus promoter. And we suspect this is because different promoters are going to have different profiles of what cell types they get expressed in. And so for our final experiment, rather than looking at any absolute transduction rate, we learned that it's important to really consider things from a relative perspective. So we can look at all these different variants across the entire hierarchy of cell types, now neuronal and non-neuronal, in a single animal. And we can score relative to each other which variant does better or worse. And we see all the same biases that we saw before. So CATB10 preferentially targeting neurons, PHPV1 preferentially targeting vascular cells specific to endothelial. We see the lack of transduction across all the different variants in pericytes. And we also see the lower transduction across all variants in these VIP neurons. So this was really cool that we reconfirmed all of the hypotheses that took many experiments and lots of time to do. And we were able to confirm it in a single animal. And even though there is this bias from UBC M-cherry that it has lower expression and a vascular bias, that we can still recover the specific relative biases of variants to each other. So the last kind of important take-home lesson is that this fraction of cells that are transduced, it's biased by your cargo, it's biased by your cell type distribution. And so we really think it's important that if you want to say something about a vector independent of the cargo that it's being delivered, that you really want to report relative rankings within an animal instead of making any absolute claims. And we just confirmed this endothelial cell versus parasite finding across all the different fluorophores and cell types, and also this VIP positive neuron, and also a reactive astrocyte finding. Both of these, all of these are cell types that seem to get missed by the variants that we tested in our study. And this is important for folks that might be working in these spaces. So in summary, kind of the things that I really wanted people to take home is don't trust off-the-shelf solutions for your use case. It may work. Many of the things out there might work, but don't assume that it will. And make sure that you double check the output of things like the Cell Ranger pipeline and even empty drops pipelines and marker gene identification pipelines. And know your assumptions. And relative is greater than is better than absolute in a lot of cases in biology. And I think as a general paradigm, I like thinking about things that way. And there is lots of room to grow, or lots of challenges still in single-cell RNA-seq data that have to be figured out for new applications. So it's a really exciting space. And then most importantly, these AAV tropisms that have been reported and that we are finding, they are complicated, they're interesting. We don't just have a variant that hits the whole brain. We have variants that hit all sorts of cell subtypes. And these things depend on many things. It depends on your capsid, depends on your cargo, your delivery method, your promoter, your regulatory elements that you add. All of these things are incredibly important to understand when choosing your vector of interest for gene therapy. And so with that, I just want to say thank you again to Michael and Tatiana for we've been in the thick of this for years now, and we're finally surfacing. We finally put the paper on bioRxiv, and Yeah, it's great, and thanks for all your help. And then thank you to all the people who along the way have helped me. I was brand new to biology just 4 or so years ago, and I think this would have been an incredibly difficult, if not impossible, project for me to do without the guidance of the expertise in the Gradinaro and Thompson labs. And that's a wrap.